morning students. Today our topic is, as you can see, Mendel DihybridCross. What is Mendel DihybridCross? Mendel, first he considered one character at the one time. Then after doing the experiments, then he considered two characters at the one time. So DihybridCross means what? A breeding experiment considering two characters at the same time. Mendel considered which two characters at the same time. These were, for his experiment, he considered one character was the seed form and another character was the color of potato. In seed form, he took the two characters, that was round and wrinkle. These contrasting characters were there in the seed form, and in color, the contrasting trait was yellow and green. Here, the round showed the dominant trait, while the wrinkle was recessive. Yellow was dominant and green was recessive. He set this dominant character and recessive character on the basis of monohybrid cross. In those seeds, the character was appearing more, that was the dominant, and the character that was appearing in the last seeds, that was the recessive character. So, for his experiment, he took the parents as pure form. One parent was round and yellow, another was wrinkled and green. So, for that we use the value capital R, capital Y, and for wrinkle, small r. These were the parents of Mendel's DihybridCross, and here you can write, so these are our parents. And here the gametes are formed. What kind of gametes are formed? It has only one capital R and one capital Y. And here the gametes can have only small r and small y. Small y, this one. Okay. Then he crossed the parents and get the F1 generation. In F1 generation, all the seeds were having dominant character, showing the dominant character. The phenotype was all were round and yellow. This was the genotype and this was the phenotype. So after that, he did selfing of the F1 generation. For selfing, he took the same combination of the F1 generation. Here, the gametes are formed, can have different combinations, like these four combinations can appear in one parent and also same in the other parent. So, these gametes were formed in the F1 generation, and after selfing, they can combine to form 16 type of combinations. Of these 16 types of combination, the ratio was obtained, that was 9:3:3:1 for these 16, in which 9 plants that were having both the dominant traits. And 3 having single dominant trait. And 1 that was having both recessive traits. we can see the different combinations of the F2 generation. This goes our F2 generation. If we want to see the genotype of these F2 generations, we can use Punnett scale. So, to make a Punnett scale, first you have to show the gametes. Here you can say male and female gametes. There are the 4 types of gametes as you see in above and 4 types of gametes below. So in male gametes, the combination, the gametes were these, and same in the female, the gametes were these. So by doing Punnett scale, you can make the different combination. Like this, in this boy cells, these 2 traits will come. So you can write as both capital R and both capital Y. Same in the way. You can write complete chart. You have to see the both lines of the gametes and make the combination. So here you can see the 16 combinations of the genotypes that Mendel obtained in his DihybridCross. Here you can easily interpret that by making this line. Then one triangle is formed. These oval you can count 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 are all having both the dominant trait. Allele having both traits, the allele dominant is present. Like capital R and capital Y are present in all these 9. While here you can see in 1, 2 and 3, only capital R is present and small y is present. Then you can see in this inverted L shape, small r in these 3, small r and capital Y is present. While in the only this one, there is only one C having small r and small y. Both were recessive. So in this way you can see 9 out of the 16 seeds, if they were 16 combinations, 9 of them having both dominant allele and the 3 and 3 having one and another one dominant and one having both recessive allele. So what was the conclusion of the DihybridCross? Here, Mendel concluded 2 points. Number 1, there were 4 combinations appeared. Another, the new combination out of these 4, 2 were the parental combination and the 2 were new combination. So new combinations appeared. Like capital R and capital Y, small r and small y. These were the parental combinations. While capital R with small y and small r with capital Y, these were the new combinations. So from here he proved, or he stated the new law, that was law of independent assortment. What does that law of independent assortment say? Here, as you can see, the cotyledon color and the shape or the form of the seed both are not stick to each other. They get separated independently in the form of gametes. So you can get, that is why you can get the new combination, because they are the alleles of the different trait, they get separated. So law of independent assortment states that the law of independent assortment states that the factors of each character are distributed into the gametes independently. To make the new combinations. So this was the Mendel dihybrid cross, and you can revise it as dihybrid cross ratio that was obtained by Mendel was 9:3:3:1. That ratio he was detected by calculated the all seeds. Total number of seeds he obtained were 556. These are the total seeds he got. Out of these total seeds, the 315, 108, 101, 315, that was showing the ratio, and the 32. From here, he interpret this ratio 9:3:3:1. That is approximately 9 multiple of these two values, 108 and 101. That is approximately nearby each other, and 32. That was explaining the one. So from here he get the ratio of 9:3:1. So Mendel did a lot of experiment on the pea plant, garden pea. First he did monohybrid cross, then he did dihybrid cross, and later in next video we will see why Mendel was get successful and why he is called the father of genetics, and what was the summary of the Mendel's experiment. Thank you.